here at Cambridge University, and he's, I believe, going to talk to us about some stochastic models for regulation of cellular proliferation.
Well, I feel like a bit of an imposter, actually. Let me start by saying that. So I'm not a mathematician. I'm not even a biologist. So I think what I wanted to do in this talk is, rather than bringing solutions, some clever solutions to problems, to invite you to look for those problems, vice versa, I think in a way I want to focus on the experiment and try to advertise some problems which I think are in desperate need of mathematicians and people who know a lot about stochastic processes, which isn't me. So the problem that my group has been interested in, well, the last 10 years or so, is trying to understand what we can elucidate about cell biological processes, particularly stem cell biological processes. And what I want to do in this talk is use one example as a kind of cameo to present the kinds of things that we're doing and why simple ideas from population dynamics and stochastic processes may be useful. And that problem is the problem of mammalian epidermal maintenance. And I'd like to mention in the talk not just examples where we have regulated maintenance of the tissue, but how these kinds of approaches can give us insights into tumor initiation and progression. Now, I know this topic is not completely alien to this meeting, and unfortunately I wasn't here for the last couple of days, but I know, for example, you would have heard, I think, from Jasmine Fu, who I think would have mentioned some of these things, so I hope there will be some resonance. So let me begin with a quick bit of background. So stem cells are defined by the ability both to duplicate, to renew themselves, and to give rise to more differentiated cell types. And there are stem cells in embryonic tissues, but more importantly for this talk, there are stem cells in adult tissues. So most of the tissues, many of the tissues in our body, particularly epithelial tissues, undergo constant turnover through adult life. You can think of blood, intestine, and so on. And those tissues are maintained by a specialist population that are able to perfectly self-renew, giving rise to themselves to maintain themselves and giving rise to more differentiated cell types. And there's a deep problem in stem cell biology, which is that these cells have to engineer something very special. They have to achieve a perfect balance between their proliferation and differentiation to maintain their overall number constant. So broadly speaking, the summary of what we're doing in my group, which is mostly theoretical and a little bit experimental now, is we're trying to understand what are the mechanisms that stem cells use to regulate this exquisite balance between proliferation and differentiation, and how do stem cells and progenitors become subverted in the initiation of disease states. So again, by way of background, let me begin with what I've mischievously called folklore. So this is what pretty much all stem cell biologists believed up until 10 years or so ago, and it's the narrative of all of the work in the field. So there's a folklore or dogma that stem and progenitor cells are organized in a very strict one-way proliferative hierarchy. It means that stem cells in these adult tissues are regulated by some extrinsic factors which we can call a niche, and those stem cells give rise to cells, progenitors, which may go through a short period of amplification before terminal division into the specialized cell types. So one bit of dogma is that we have this strict one-way proliferative hierarchy, and another bit of dogma is that the stem cells are individually long-lived. So biologists like to think that these cells undergo perfect asymmetrical division to reproduce themselves and produce one more differentiated cell type. And finally, that these cell types are defined by signature expression of molecular markers. And it turns out that these three bits of folklore or dogma, all of these are unsafe, actually. All of these are wrong in what turns out to be quite an interesting way. And I think it's an interesting, what can I say? So I suppose the punchline of my talk is it's by using ideas from stochastic processes and so on that we can elucidate the fact that these are wrong and what are the right rules instead. There are rules. So in view of this, the search for mechanisms of tumor initiation, well, they tend to emphasize the potential role of these cells that look like the long-lived cells in tissues, that these are the cells that acquire mutations and initiate tumors. And so people talk about this kind of cancer stem cell notion, the idea that that tumors are maintained by a minority population of tumor-maintaining cells. Also very controversial and also probably not correct. So are these principles really correct? And I suppose what I want to address in this talk is how can simple ideas, when I give this talk to biologists, I don't put simple here, but I'm scared in this audience. I need to put, these are super simple ideas. You will think they're laughably simple. Ideas from population dynamics. How can they provide us with insights? So we've looked at a variety of tissues, actually a very large number of tissues and organisms by now, but I really want to focus on this one. It's mammalian epidermis, and it's a kind of poster child for adult stem cell biology because, as everybody knows, this is a tissue which is constantly turned over through adult life. Differentiated cells are shed from the outer surface of the skin, and they are replenished by stem cells, allegedly, stem cells which reside in the basal layer of the tissue. And it's also a canonical stem cell supported tissue in that it can undergo rapid regeneration following injury. So here's a section through human epidermis, and it shows that epidermis has multiple components: hair follicles, sebaceous glands, and the interfollicular epidermis. And here I want to focus just on interfollicular epidermis. So in interfollicular epidermis, all dividing cells adhere to the basement membrane, and when those cells commit to terminal differentiation, they detach from the basement membrane, they rise up through the suprabasal layers, and as they move up, they express more keratin. Eventually they lose their nuclei, they knit together, and they are shed. So this is a system in which we've got constant turnover through adult life. And my background is theoretical physics, and there is an ideal tissue for the theorist, which is esophagus, which we'll also hit upon briefly in this talk. An esophagus is just interfollicular epidermis. It's basically epidermis, but with no appendages. Okay. So the question I want to address is, how do stem cells in this tissue, where do they reside? In the basal layer, presumably, and how do they function to maintain the lifelong turnover of tissue? So in outline, what I want to do, it turns out that the history of the study of this question will be quite interesting. And so I'm going to begin by reviewing rather quickly the history of how people have tried to understand what's going on in that tissue. And then I'm going to introduce this notion that we can make use of genetic lineage tracing to follow the fate of cells, of marked cells, and their labeled progeny over time, clones, and how ideas, simple ideas from statistical physics or population dynamics can help us actually to elucidate what are the rules of cell fate in this tissue. And then we'll talk about how that can provide us with a platform to understand the initiation of a particular kind of cancer in mouse, of basal cell carcinoma. And then, depending on the clock and the chair, I'd like to talk finally about how we can do in vivo tracing in human epidermis, again using ideas, simple ideas from statistical physics. And just like Ray said this morning, this talk may work better if you will ask questions. So if I say something which is alien and confusing, please do shout out. It's very much appreciated. Okay, so let me start with the history of this question, and it goes back to this gentleman. So this is Charles Philippe Leblond, and this is work that he did in the year that I was born, 50 years ago. And this actually is a very special scientist. So he's the father of modern stem cell biology, and he's also a little bit my nemesis, because we feel over the last 10 years that we made some discoveries in my group in the area of stem cell biology. And each time I really think I discovered something, I discovered that he discovered it 50 years ago, and then everybody forgot that. So he was the first person, so he understood very deeply, and he wrote a beautiful essay on that if you want to understand cell fate behavior, you have to make a dynamical measure. So post-genomics, everybody forgot that, because you just, you kill the cell and you measure the transcriptional profile or whatever it is. But if you want to understand fate, you've got to combine all of that with at least one dynamical measure of what the cell was today and what it was tomorrow. It made a some fate decision. And he understood that. And in the old days, he looked at lineage tracing in the simplest possible way, the only way they had at that time, which was to use the incorporation of tritiated thymidine. And tritiated thymidine is incorporated in S phase of the cell cycle. So if you inject tritiated thymidine, in this case into a rat, and you wait for a day or so, the cell which incorporated that can go through division, and here are two cells, the progeny of a single division. And what Leblanc did was he looked at the basal layer and suprabasal layer of rat esophagus, and he could see that some of these cell doublets were wholly in the basal layer, some had cells in the basal and suprabasal layer, and others they were both suprabasal. And he wrote in the kind of poetic way that people did in that time, the frequency of these three types of pairs was such as to indicate that the transfer of a basal cell to the spinous layer is a chance event. It's a very novel way of thinking back in 1950, a chance event which can affect any basal cell. So the picture that he had was that the entire basal layer was built up of progenitors which divide symmetrically and then essentially flip a coin to decide who will differentiate. That's what he proposed back then. Turns out that this is wrong, but actually the essence of what's really happening is actually correct here. So this was quite prophetic back in 1965. Then 10 years on came along Chris Potton and others who completely wiped away Leblanc's work, and they came up with a completely different paradigm which has not just dominated the field of epidermal stem cell biology, but actually dominated the field of adult stem cell biology across the board. So what Chris Potton proposed is that epidermis is made up of a mosaic of clonal units in which there are long-lived slow-cycling stem cells that give rise to short-lived transit amplifying cells that go through a strictly limited number of divisions before terminal differentiation, and then those cells rise up through the suprabasal layers before they're shed. So the whole tissue is built up of a mosaic of these units, which they called epidermal proliferative units. And here is a section through tissue which shows that putative unit. This will turn out to be completely wrong, but this was the model which dominated the field of epidermal stem cell biology since 1974. So all experiments done in this field are looked at through the prism of this model. And I was a kind of migrant from condensed matter physics, and to put these things in perspective, I discovered that there are more people in the field of epidermal stem cell biology than there are in my old condensed matter physics. It's a kind of scale. So all of those experiments were seen through the prism of this model. And then came along another very important, the third important landmark in this field, which was undertaken by Howard Green and Jan Barenden. And Howard Green, he was the first person to develop a culture system in which you could grow human skin. So he was the first person to make artificial skin for skin grafts. And what they noticed was that if you took primary human keratinocytes and plated them, there are three types of colony. One of them grows bright, shiny colonies which can be serially repassaged and can produce human skin. And another set of colonies grow small colonies which terminally differentiate, and something in between that they called meroclones. Three types of clonal keratinocyte types. And this matched very nicely with this idea of stem transit amplification. And there was further evidence in support of that based on molecular markers and adhesion and various other things. So after that work in the mid-80s, the stem transit amplifying cell hypothesis became the prevailing model, and Leblanc's early work was completely forgotten. But is it correct? And of course it isn't correct, otherwise there wouldn't be much of a talk. So how can we do, now in the 21st century, how can we trace the fate of cells? And how can we really target this question about what are the rules of cell fate in this tissue? Well, these days there are much more refined techniques, and the one which you're going to see a lot more of in the magazine journals more and more is intravital live imaging. So these days, it's possible to trace in real time the dynamics of cells in living tissues. But this is still quite restricted. Maybe here you can trace the fate for a few days, but you certainly can't trace it over the lifetime of the animal. So the second approach, and one which I need to describe before we get to a little bit of analysis and math, is this one. So I don't know how many of you have heard of this so-called Kree-Lox system, and so I'll just run through that. So the other way of tracing the fate of cells is to mark some of those cells with a label and then look at what they've done over some time interval. And the standard technology for doing that is a transgenic approach where you introduce a fluorescent reporter gene into the genome, but you place that downstream of a stop cassette, so it's not transcribed. But the clever bit is that the stop cassette is between two sites which are known as LOXP sites. And those LOXP sites can be excised in the presence of what's known as a Cray recombinase. Right? So the idea is that following expression of a Cray recombinase, you can excise the stop cassette and then you get a fluorescent protein. Now, if you put your recombinase under the control of some promoter of your choice, then what happens is that the Cree is driven off that promoter and it lives in the cytoplasm and the estrogen receptor. But when you add a small dose of tamoxifen, the Cree is released into the nucleus, it excises the stop cassette, and then the cell then has a hereditary label which it will carry for its whole life, and it will pass on to its progeny. So the idea is that at a time of our choosing, we can label a targeted set of cells individually, and then later on we can go back and ask what happened to the cells and its progeny, which is called a clone. So this is what we did. Back in 2007, in collaboration with Phil Jones, we adopted this genetic lineage tracing approach using a ubiquitous promoter. So what would we expect? We label some tiny fraction of cells in the tissue, and if we label a transit amplifying cell, then we'd expect it to divide a few times, detach and be lost, stratify and be lost. But if we labeled one of these long-lived putative stem cells, we'd expect it to grow some proliferative unit, some epidermal proliferative unit. And here is a confocal section through the basal layer of mouse tail epidermis, showing an ensemble of clones. You have to kill the animal, of course. These are, of course, different clones, but they're different clones. Here at 1 week, we can see a clone with one basal cell, and here at 2 weeks, there's a clone with 2 basal cells. It might have multiple suprabasal cells, and here at 3 weeks, there's one with 4. So this is like its footprint, the clonal footprint on the basal layer. So what we did, or we got a postdoc to do, was to simply count all of those clones. And so each of these dots is a particular clone which had a certain number of basal cells. And what we saw, to our surprise, is that the average size of those footprints, the average size of those footprints over time, instead of rolling over, as we'd expect for such a model, actually goes on increasing, and actually it goes on increasing linearly with time. And the distribution of clone sizes becomes increasingly broad, as you can see here. Okay, sorry, I think there was a question.
So, sorry. Can they apply tamoxifen to specific cells, or is it just a random subpopulation of cells that takes up the tamoxifen?
Yeah, so it's a great question. So the tamoxifen, you just inject it into the animal, but the Cree is driven off a promoter. So all the cells that have that promoter could be induced to express this yellow fluorescent protein. So if you use one which is epidermis specific, you only get cells there. If you use one that's ubiquitous, you'd get them everywhere.
Oh, okay. So you're going for, so it's a tissue-specific promoter then?
It's up to you. We use ubiquitous ones, we use targeted ones, different ones. Good question, okay. All right, so what's going on here? What's going on here? In principle, we have high complexity. There are different clones making different fate decisions. But, you know, I'm a bit physicsy, and one of the things that we notice very quickly is although these distributions of sizes are very broad, in fact, if you scale them by their average size, then all of these data sets collapse, and they collapse onto something like an exponential size distribution. So in other words, we have a broad distribution of clone sizes, but we have a scaling phenomena which says that although the average size of the clone is increasing, the chance of finding a clone of some multiple of the average, that remains constant over time and defined by an exponential distribution. Right? So that's good because it means that actually there's one rate limiting process in this system, and hopefully this should translate to some simple stochastic dynamics. So what's the origin of scaling? And the answer actually is very simple. So in fact what's going on in this tissue is that we've got progenitors that can duplicate, they can terminally differentiate on division, or they can asymmetrically divide. But we have to maintain the overall number of progenitors constant, so the chances of duplication must perfectly match the chances of differentiation. And then these differentiating basal cells, they can stratify. So this is my one-step master blah, 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 right? That you know very well. So if we look at individual clones, we can see that by chance we could lose a clone through differentiation, and by chance we could have a clone that expands so that the overall population remains constant. And this isn't a very original problem. And of course we can think of what's happening in the clonal record as giving each of these cells a surname. And the progenitors are like fathers having two sons, two daughters, or a son and a daughter. And of course this dynamics is just the dynamics of famously, of the extinction of family surnames. That's what's going on in this tissue. And if we just focus on the progenitors, then we're down to a problem where even I can do the mathematics, a critical birth-death process. And so if we ask what happens in this critical birth-death process over time, the average size of surviving clones, that forever expands linearly with time. The chance of surviving drops like 1 over time. Sounds right, because the product of these better be constant. And if we look at the chance of finding a clone with more than n progenitors, then it's precisely the scaling form with the exponential size distribution. Well, if you want to put the differentiated cells back, the math becomes a bit more unpleasant and actually more difficult than I can do. But Tibor Antal and Krapivsky actually solved that problem explicitly back in 2010. So we can go back to the original data, and well, it's noisy data. The experiments were challenging after all, but in the end we can actually get a very good fit on the back of this very simple-minded dynamics, that we have a single population of progenitors that divides stochastically at this rate, where 1 in 5 divisions lead to loss and replacement, and the other 4 out of 5 lead to asymmetrical division. Yes?
Is it really a one-step process, or is it that the stem cell divides into 2 and there's a rate of commitment differentiation?
Yes, that's right. So good point. Yes. So if we look at this dynamics, we have cell division giving rise to basal differentiated cells, and those basal differentiated cells then have a timescale to stratify, which is more or less the same as the division rate, of which we can also infer. So needless to say, this was an incredibly unwelcome contribution to the field, and everybody hated us, and still do, actually. And there were many criticisms we got, though maybe this one, you know, so the last refuge of a scoundrel was they were saying, well, maybe Mao's tail is strange. And so my collaborator tortured a poor student to repeat all of these experiments in the tissue for which the EPU concept was introduced, this proliferative unit concept was introduced, which was mouse ear, and we get exactly the same thing, more or less. It's a slower division rate. And here you can see those putative epidermal proliferative units, and here you can see the clones projected expanding underneath them, and they don't care about these boundaries. And what about esophagus? That was the problem that Leblond had considered. So if you look at esophagus, we also find very similar ratios. And we can ask, have we just rediscovered what Leblond found? That's a good question. And I think not quite, because if we go back to what Leblond proposed, then under this kind of dynamics, we'd expect to get, if we interpret it this way, it's like flipping 2 coins and getting heads, heads, tails, tails, or head, tail, tail, head. So we would expect statistics 25%, 50%, 25%. And the fact that these are different suggests that the fate outcome is correlated. It's not a uncoupled random process. Okay, so should we be surprised? Well, we were. But at that time, this work was done by an extremely smart graduate student who's now on the faculty at Harvard Medical School, called Alon Klein. And Alon was interested. Actually, Alon is a very interesting guy in many ways. He joined my group. In those days we worked on cold atoms, and he wanted to work on cold atoms, and he got seduced into this problem of looking at epidermal maintenance. And then when he finished his PhD at ridiculously early, he then went to Mark Kirshner at Harvard Medical School, and Mark had him doing experiments on arrival. And now Alon has invented this droplet sequencing approach that's become an industry, and this is this same young guy. So Alon was extremely smart, and Alon came into my office one day and he said, you know, we should have really expected all of this. And the argument was very simple. He said, if we have a tissue which is maintained by an equipotent cell population, it means that maybe these cells are making stochastic fate decisions, but they have the same regulatory program. They've got the same set of dice, if you like. And of course, that's a condition of homeostasis, because if we had cells which were forever clonally fit and other cells which are unfit, then one set will take over, and that's not homeostasis. So if we have this equipotent population, then there are only three patterns of long-term clonal evolution that are possible. We either have invariant asymmetrical cell division, or we have asymmetry enforced at the population level, so that some cells might duplicate, others might terminally differentiate, others might asymmetrically divide, but the balance is enforced at the population level. And within that, we've got two possibilities. Either this balance is enforced cell autonomously. It means these cells exquisitely balance proliferation and differentiation without being conditioned by the environment. A critical birth-death process, after all. Or we have cells which are sensitive to the environment that duplicate in response to the loss of neighbors, or vice versa. Right? So think of a voter model would be a good example. And so what he said, which is what one can show, is that however complex is the pattern of self-renewal, long-term the clonal evolution will be indistinguishable from one of these three universality classes in an RG sense. So put differently, we can divide everything up into four classes according to whether balance is engineered at the level of intrinsic or extrinsic regulation, or whether we have invariant asymmetry or asymmetry enforced at the population level. An epidermis appeared to wind up in this category. So how do we know which class? And if we focus on the interesting set, which is population asymmetry, then in both cases we have this process of kind of neutral dynamics. We have clones which expand in response to the loss of others. And if we look at the size distribution, in both cases we predict that we will have scale invariance of the distribution. It means the chance of finding a clone larger than some multiple of the average will remain constant, even though these clones expand over time. But we can discriminate between these classes on the basis of what is the scaling function and what is the average size dependence. So in particular, if we're dealing with a critical birth-death process, the average size of surviving clones is linear. We have an exponential distribution. If we're looking at extrinsically regulated systems, then we have a different scaling function and a different growth characteristic. And these models are likely to be more familiar to many of you than they are to me. So this is just a critical birth-death process. In one dimension, we have just a coalescing random walker, and in two dimensions, we have a 2D voter model or Moran process, things that many of you will be much more familiar with than I am. What's a bit annoying is for extrinsically regulated in two dimensions, we still get the same exponential distribution, and the only difference between critical birth-death is the fact that we have this two-dimensional logarithm in the average growth characteristic, but logarithms are always, of course, hard to track down. So what about experiment? Does this work? It evidently worked for the cell autonomous case. Does it work in general? And that's been a history of the last sort of 10 years in my lab, which is that every single tissue that we look at, which is turning over, falls into these classes. So here is posterior midgut, which is a quasi-two-dimensional epithelium, and that falls onto this 2D voter model class. And the intestinal crypt is actually, curiously, it's like a one-dimensional system, and that falls onto this class of coalescing random walkers. Here you can see several time points and the scaling distribution. And these are very attractive. I mean, I advertise this because I think these are very attractive systems for people who are good at mathematics to actually dig into, because there's real data here and there's really interesting questions. And if you look at this problem of intestinal crypt, to give you an idea of the kind of resolution that they have there now, then here is a multicolored labeling system where you create a mosaic, and these are sections through the base of intestinal crypts where you can see this coarsening process that you get in a coalescing random walk. The idea is that these are all stem cells around this annulus. You lose one, you replace it by its neighbor. And this is data. This is experimental data. So there's very beautiful experiments out there which are really calling for some analytical insights. Okay, well, let's get back to skin. So we had deduced that we have a single equipotent population that maintains tissue through stochastic fate behaviors. And I said something which was a bit surprising, which was that the regulation of this balance was cell autonomous. Now, if you're a physicist, and many of you are, then actually this is kind of not really possible. In 2 dimensions, if you had cells making these decisions stochastically in a cell autonomous way, you actually get logarithmic fluctuations in the density. So it can't quite be fully cell autonomous. But let's see to what extent it is. And the other question is, what about that Barrandon Green culture assay, which seemed to show proliferative heterogeneity? Right? So what about this one? In the culture assay in human, we got these 3 types of colony. If we say there's just one population, what are these 3 colony types? And to answer both questions by an observation, what we looked at relatively recently was plating primary human keratinocytes in culture and then filming these cells over time. So here's one of these keratinocytes here, and it will divide. These are all feeder cells in the culture dish, and it will go through a round of division. And Amit Rushan scored a lot of lineages by tracing the dynamics of these cells, the clonal dynamics over time. And he could see colonies where you had symmetrical divisions, largely, putative holoclones, but he could also see these paraclone colonies where you had division and differentiation. And if you look at the ensemble of paraclones, then you find these statistics. So in other words, these cells, at the level of single cells on plastic, are making balanced, within error bars, balanced stochastic fate decisions in culture. Right? So it's a strong indication, it doesn't prove it, but it's a strong indication that this balance between proliferation and differentiation is actually engineered at the level of single cells. How that's done, we have absolutely no idea. But what about this population? We have another population that seems to function as in this stem-like behavior with these symmetrical divisions. What are they? And of course, it's very natural to ask, maybe mouse has a hidden, very, very, very slow cycling stem cell population that of course we would never see. If it just sits there and doesn't do anything, we actually would never see it. And so let me be relatively quick with this. Okay, so really that's the main point of what I wanted to convey. So now we can dig a little bit more deeply. And this gentleman, Cedric Blancpain in Brussels, he did a lineage tracing study in skin using a targeted promoter system using involucrin and K14. And K14 is enriched in the basal layer, and involucrin is enriched in the suprabasal layers. And he traced the fate of cells over time in that system. And what we found together with him, was that actually K14 indeed preferentially targets a slow cycling stem cell population. So in fact, in this tissue, what we have are cells which asymmetrically divide largely, which are very slow cycling, and we've got these cells that follow balanced stochastic fate. Why do we host 2 populations? And the speculation at the time was that one population was responsible for repair in crisis, their slow cycling had become active, whereas this is the kind of workhorse of tissue turnover that engineers the maintenance of tissue. And in esophagus, we never see that population. All right, so this gives us a platform now, a quantitative platform, to begin to understand what may happen when we have a transition to dysregulated growth and tumorigenesis. So this now is a very popular activity in the field. There are zillions of labs who are doing this kind of thing. So what you can do with this same inducible labeling system is not only can you induce a fluorescent protein reporter, but you can also induce an oncogene. So the system that we chose to target was hedgehog signaling, which I don't have time to go through. But in short, if you activate smoothend, which is part of the hedgehog signaling pathway, then what happens is that you transit slowly from normal type of behavior to a hyperplasia, hyperplastic growth of the clone, which then becomes dysplastic. It starts to embed itself into the epidermis, and eventually, after a couple of months, it transits into invasive basal cell carcinoma. So we have our quantitative platform that we can begin to ask what are the processes that occur when we activate smoothend in this system from, if you like, population dynamics point of view. We know what the normal rules are. Can we elucidate what the rules are under these conditions, or whether there are rules at all? So if we do that, we can induce under the control of the involucrin promoter, which targets the progenitors, or we can induce under K14, which targets the slow cycling stem cells. If we induce under the involucrin promoter, we find that we tilt cell division towards proliferation. But what's interesting here is that there are still rules, right? So these cells still function as an equipotent population, but now they're persistently imbalanced. That's not obvious at all. It could have been that all the cells did something different according to their environment, but they function as though they are still a single equipotent population, tilted towards proliferation. And these would just grow out and grow larger if it wasn't for the fact that the division rate is falling. And if we look at what happens if we induce on K14, again, we have this tilt towards proliferation, and they also get bigger. So in both cases, we have this constant fate bias towards proliferation. But are both the population stem and progenitors, are they both competent to induce invasive basal cell carcinoma? And because I'd like to have the last 5 minutes for a different topic, I'm going to give you the punchline, which is that although both are tilted towards proliferation, actually only the stem cell population progresses into invasive basal cell carcinoma. So we can follow that dynamics, and it's only the basal population which has the capacity to generate invasive tumors long term. All right, so let me, I think I have 5 minutes or so, don't I? So let me give you a sort of intermediate summary, and then I can get to the last part of the talk. So maintenance of mouse epidermis involves the stochastic fate of a single progenitor cell pool. The lineage tracing using targeted promoters shows that we have a sustained proliferative hierarchy in epidermis involving a very slow cycling stem cell and an almost balanced, within resolution, balanced progenitor population, where it looks like the function of this population is to engineer repair and this to engineer normal homeostasis. And following smoothend activation, both the populations have a sustained proliferative advantage. They function as an equipotent population, but only the stem cell fraction appears competent to initiate basal cell carcinoma.
I think I just missed one of your points, which is I thought that without the smoothing, you had some sort of balanced state, but you have the green and the red unbalanced. What is that lack of balance in the right supposed to indicate?
Yeah, so what this is supposed to indicate is that this is a slow cycling population, but it's still cycling very slowly. So there is a super small imbalance there. All right, so in my last 5 minutes, I shall get through the following, which is how can we do in vivo tracing in human epidermis? And why would we want to do that? And the reason that we'd want to do that is that humans are in many ways different to mice. I mean, they're different from the perspective of longevity, but actually more importantly, the clonal activation of an oncogene is not actually very much reminiscent of exactly what happens in a tumor initiation process in normal human tissues. So can we develop a lineage tracing approach for human tissues? And of course we can do that, but you'll instantly spot the flaw. Of course we can do that because we've got a clever way of marking cells, because on DNA replication, cells of course spontaneously acquire single nucleotide polymorphisms. They acquire point mutations. And those point mutations, if they occur at a low rate, are a clonal marker. Those point mutations are conferred onto their progeny. And so in principle, we can use deep sequencing approaches to resolve the density of specific mutations in biopsies. And from that, we can infer the sizes of mutant clones, and we can infer something of the dynamics. But there's an obvious problem here, famously, which is that these are, after all, mutations. So we could suspect that these mutations may not be neutral at all. It may not be a neutral marker. It may actually change the dynamics of the cells. Well, let's keep a good thought and let's have a look. So last year, middle of last year, there was a paper came out. Actually, this is the same Phil Jones that I was collaborating with in the past, and Peter Campbell. There was a study that came out from the Sanger lab that got a lot of publicity. And they did something which in hindsight was completely obvious, but surprisingly not many people were doing that. They decided to do exome deep sequencing on physiologically normal skin or normal tissue. So everybody is studying deep sequencing of tumors, but they said let's just have a look at normal skin. And it's a bit unpleasant, I admit, but this actually is a kind of cosmetic surgery byproduct of an eyelid epidermis. So these are eyelid epidermises which are taken from middle-aged patients where the skin has grown a little bit large and they want it uplifted, and this is what is removed. And they took biopsies from this tissue and they did deep sequencing on that tissue. And they came up with a very striking conclusion from that study, and it made our local news over here, and it was on NPR and everything else, because what they found was two things. First of all, the mutational landscape of this physiologically normal skin overlapped almost indistinguishably with squamous cell carcinoma. These patients had no carcinoma. But they also deduced, remarkably, multiple cancer genes are under strong positive selection even in physiologically normal skin. So I think earlier in this meeting you heard about field cancerization, I think, or anyway, many of you know what that is. So the idea here is that these cancer genes acquire mutations, they acquire a clonal advantage, and they rip through this tissue, preparing the tissue as a kind of pre-neoplasia. So this was big news, right? We all should panic because we're carrying around all of these clones which are poised for transformation. But is it true? Okay. So what's the idea here? Well, I already said it in words. You can do deep sequencing. They did deep sequencing. And what matters here is this so-called variable allele fraction. So for different mutations, you can identify the frequency of those mutations in a biopsy. And if you multiply that by the size, these are heterozygous mutations, so a factor of 2 for the alleles, then we can get the clone area. And here's the frequency of clone areas from their data. And here you can't see, but there's a long tail here. And these are these vicious clones which are taking over. But I was slightly suspicious by this study, because if you look at the average clone size for all of these vicious mutations like Notch1, P53, and so on, they were only a factor of 2 larger than the baseline. These clones have had 50 years of advantage, and all they can end up with is a factor of 2 increase in their average size, these vicious clones that are taking over. So why is the proliferative advantage so small? Well, what happens if the mutations conferred absolutely no proliferative advantage whatsoever? Well, now we're in business, because it's the same old, right? What we can do is we can go back to our inducible labeling system. But now the situation is slightly different. It's not a critical birth-death process, but it's a critical birth-death process with immigration, because you can create these clones, point mutations, at any time, and they can expand a bit and contract, and most of them will be lost. And that leads to a size distribution, chance of finding clones of size n, which has this form. Even I can do this math. So here we've got a 1 over n, and that 1 over n comes from the fact that most of your clones were born yesterday in this process. Because you receive them, there's neutral drift, and then they disappear, so the majority are born in the relatively recent past. And if we take that size distribution and we build the first incomplete moment, then we would predict that the first incomplete moment, the chance, this quantity, should just be pure exponential. Well, that's the theory. Let's look at the experimental situation. Oops. So here are the clone sizes for all the mutant clone IDs of a particular patient, this one, which span 92 biopsies. And some of those point mutations belong to a single biopsy, others belong to 2 of the biopsies, others belong to 3, and so on. And in this patient, there's one point mutation which appears to belong to 5 biopsies. So let's keep things safe to start with. Let's look at the ensemble of synonymous point mutations. So the synonymous ones are ones that don't change the protein sequence, so they're probably going to be neutral. So those are the red ones here. And here's the size distribution for those synonymous mutations. So what do we predict? We predict that the first incomplete moment of this distribution should be pure exponential. Well, it's noisy data, but it isn't bad. So this is consistent with neutral clone dynamics. But what about the non-synonymous mutations? What about those? And here we've got many more. Here are the distribution of non-synonymous mutations. And if we look at the first incomplete moment, it's pretty exponential. But it isn't quite. There are some outliers here. Right? So maybe these outliers are important. Let's look at those. And if we look at those outliers, this deviation from exponential is associated with just 7 mutant clones. So we've got 1,400 mutant clones here, 1,300, but there's just 7 that are responsible for this deviation, and they belong to 6 biopsies. And if we look at those biopsies which give rise to these outliers, they contain variable allele fractions. So this is the outlier. It's a mutation, a point mutation in MML2, whatever that is. That's the outlier. But you see that that biopsy has other mutations in other genes with a very similar allele fraction. And almost certainly that is a clone, a single clone that bears all of those point mutations. The point is to generate a non-neutral transformation, a single point mutation, heterozygous point mutation in the gene is not enough. You need mutations across a range of genes. You need epistasis. If we remove those 6 biopsies, then we get very good exponential distribution. And here you can see a deviation back in the probability distribution. And this is just the resolution limit of the deep sequencing. So I'm basically done now. And so, okay. So, so what about mutant clones that span more than 1 biopsy? Oh yeah, we can also dig out the loss replacement rate out of this data. It's about once per month, which is reasonable. So in situations where we have clones spanning multiple biopsies, indeed they do have this characteristic of having multiple mutations across a range of genes, and we can deduce the order in which those mutations are required. And here's other patients. It works extremely well. So exome deep sequencing actually gives us a quantitative means to probe stem cell self-renewal in human tissue. It will work not just in epidermis but in other tissues. And it gives us a quantitative means to detect and characterize mutations, rare mutations, that are associated with field transformation and their mutational signature. The vast majority of point mutations leave this homeostatic, balanced, stochastic fate unperturbed. And of the rare mutations, the rare mutant clones that lie outside this neutral distribution, most have mutations across a range of genes. Right. So, okay, so the perspective, right? So we can find evidence for and against proliferative hierarchy, and how could these observations be reconciled? So what's really going on? And I think what's going on is that we have a niche Signals from the dermis, for example, may promote quiescence of this population and may confer proliferative advantages, local signals from the niche. I actually don't think that there are really two populations. I think there's just one. And under normal conditions, cells can exchange between these two groups. But when you transfer these cells to culture, I think you can lock cells in these states, which is why I think we have this holoclone, paraclone type dynamics. That's what's really going on. And oncogenes can act differentially on these cell types. So my last slide is the following. We started with this folklore, one-way proliferative hierarchy, individually long-lived, signature expression of molecular markers. And actually what this study shows and other studies show is that stem cells are actually frequently lost and replaced. They're not individually long-lived. And what I didn't show you, but in other contexts, is we can see that those cells can move reversibly between states prime for proliferation and prime for differentiation. This is much more of a continuum than people imagined before. So I'm done, and I think I thanked the experimentalists as we went. I particularly wanted to thank Alon Klein, and actually the cancer work was really the new work, and that was done by a postdoc of mine, Eduard Henezo, and the experimental work was done in Cedric Blancpain's lab, Adriana Sanchez. Thank you.
All right. Questions? So one question that occurs to me is, especially if one tries to think about trying to make some, get some mathematical or physical understanding of what's going on. It sounds like the analysis you've done so far is largely at sort of, and I don't mean this in a critical way, but in a phenomenological rather than dynamical sense, like, okay, is this picture for what's going on with proliferation consistent with the data, and then you do some sort of statistical tests on that? Do you see there as being the potential for more mechanistic models to underpin these phenomenological models, or do you think this is kind of the best that can be done given available data and the complexity of the biological systems?
So this, of course, is a great question, and it's a kind of philosophical question about how we approach biology. So one question is that we can try to get all the possible ingredients that we think are key from some gene regulatory network, extrinsic factors, and try and build a model which can then predict and describe what we see. And the approach that we've taken is a very different one, that we ask whether there are hallmark properties of the experimental data that inform on what is the simplest possible canonical model that could explain that. And then use that iteratively to dig deeper into the regulatory programs that operate. That's exactly the strategy that we use. And actually in the case of epidermis, I think we haven't been very good at that yet. So it's obvious, for example, that extrinsic mechanical factors play a role in regulating these fate decisions. They may have simple stochastic outcomes, but the underlying regulatory program involves mechanical cues. But there are other problems that we've looked at where we have done that. And to just sort of illustrate that with an example, so one of the places where we have this process of loss replacement of a voter model-like dynamics is in the germline, the maintenance of male germline, spermatogenesis. But what we backed out there, because we have much more exquisite data from live imaging and from molecular profiling, is that what we realize now is that cells feed off a niche. It's like quorum sensing. They feed off the niche and they consume a niche factor that maintains stemness. When they move away from that niche, they begin to downregulate stem cell genes and upregulate differentiation genes, leaving those cells competent to differentiate but not committed. But if they get back to those niche factors, then they're able to switch back. It's actually my cartoon, here, this kind of notion of priming. And there we can build the next step of more refinement. So there we're describing this process of heterogeneity in terms of dynamical theories, because we have the data to challenge that, right? So my philosophy is not to outreach the data. I want the model to be as simple as possible, but not simpler.
Top down from the data.
Yeah, exactly. I mean, and anyway, I mean, it's my strong view, I'm not alone in this, but my strong view is that if you're working in biology, there is so much data out there. You should start with that, in my view. You know, I come from condensed matter physics where it's the opposite way around. It's a paucity of data, so theorists are challenged because they've got to try and think of new phases of matter with no clues. But we're living in an era where the real bottleneck is in the fact that there's this tons of data and very few ideas about what to do with that. And I think, you know, starting with the data has been a successful strategy with this, and I think will be successful in other contexts.
All right, so let's thank Ben one more time. Oh, we're just in time for